Hello, everybody. Thanks for joining me for today's webinar on enzymes and anaerobic digestion. I'm Dan McKeaton. I'm a chemist and senior researcher at Aquafix Inc. You can reach me at dan.s@teamaquafix.com. I've worked over at Aquafix Laboratories for about 10 years. At Aquafix Laboratories, we do a lot of research and development for Aquafix products. We also provide consultation and some lab services for clients. Lately, I've been focused heavily on getting anaerobic pilot digesters to work so we can do a better job studying site-specific treatments in anaerobic digesters. Feel free to reach out to me if you have any questions about our lab services. So today's presentation, like I said, is on enzymes and anaerobic digestion. So this is just a brief layout of what I will be going through in the presentation. We're going to start with a little overview just to make sure we're all on the same page as to the content we need to know for the presentation. Then we're going to talk about key concepts like hydrolysis versus metals limitation systems and how enzymes work in general. We'll talk about the roles of enzymes in anaerobic digestion. So what phases of the treatment are they significant in? And then are enzymes effective at improving anaerobic digestion? We'll answer these questions looking at a combination of academic literature and internal studies. So here's an overview of what we're going to be going through on digesters, starting with how the process works, key parts to the anaerobic digestion process, and then we're going to talk about nutrient requirements, which are heavily tied into enzymes in anaerobic digestion. Anaerobic digestion takes place in 5 steps. It starts with disintegration, then it moves on to hydrolysis, acetogenesis, acetogenesis, and methanogenesis. Disintegration tends to vary pretty widely from site to site in anaerobic digesters. This is the process where large solids clusters are broken into fine particulates. This increases the surface area of these compounds, allowing microbes to get a better reach on them and make more contact. So it's increasing their surface area to allow hydrolysis, the next step, to take place. Yeah, there's wide variability with disintegration in anaerobic systems because in a lot of cases, you have, a UASB type system wouldn't have any solids going into the system. You'd have a really low solids flow, so disintegration would be pretty much irrelevant to it. If you had solids going into your UASB, you probably would just mess up the granules anyway, so we don't want to go there. Complete mixed digesters tend to do the best job with disintegration because they have continuous mixing suited for high solids content to break all those solids down to maximize digestion efficiency. Something like lagoons would be very inefficient from a disintegration standpoint because almost all of the mixing in most lagoons is just the inflow going into the lagoon. Doesn't really have a good opportunity to break things up. This is also relevant in the enzyme usage we'll talk about later on because mixing helps things bump into each other. Enzymes need to bump into substrates. If this doesn't happen, the enzymes don't work. So you have to pay extra mind to that in lagoons. Talk about that more later in the presentation. So the next phase is acetogenesis following hydrolysis. Acetogenesis is the process where substrates that have been hydrolyzed, carbohydrates, amino acids, and long-chain fatty acids, as you'll see in the diagram to the right, get converted to acids, which are then converted to mostly to acetate by acetogenesis, so acetate-producing microbes. There is also hydrogen and carbon dioxide produced in a byproduct of these steps, which is also relevant to later in the digestion process with methanogenesis. Now, methanogenesis, you're using acetate and hydrogen and carbon dioxide to produce methane, and you have some residual carbon dioxide at the end. Methanogenesis is often the key rate-limiting step in anaerobic digesters. This is taking part by 2 separate groups of methanogens, the acetoclastic methanogens, which produce methane from acetate, like we just mentioned, and hydrogenotrophic methanogens, which produce methane from hydrogen and carbon dioxide. So these 2 communities, they're kind of interesting in how they interact. You don't want to have an accumulation of hydrogen in an anaerobic digester. It tends to inhibit the acetoclastic methanogens. So these 2 communities are complementary. Usually the acetoclastic methanogens are producing the vast majority of the methane generated in a process through metabolism of organic carbon, but the hydrogenotrophic methanogens can in some cases produce more depending on the waste substrates entering into the system. The other thing which has been of note in some of the studies we've been doing lately on hydrogen sulfide, Sulfur-reducing bacteria tend to compete a little more directly with hydrogenotrophic methanogens than acetoclastic methanogens. So the acetoclastic methanogens, more or less, if they get their carbon, they're happy. They can coexist peacefully with sulfur-reducing bacteria. And then you get a splendid environment where you have quite a bit of methane being produced, but it's also all terrible quality because your hydrogen sulfide, or the sulfur-reducing bacteria, are just spewing out a bunch of hydrogen sulfide that you have to remove to be able to make use of that biogas. So that's bad. Generally, the hydrogenotrophic methanogens can help to compete with the sulfur-reducing organisms to prevent this sort of thing from taking place, improving methane quality overall. So nutritional requirements are a big key to anaerobic digestion. You'll notice looking at the slide that if you compare this to something like an aerobic process, the carbon to nitrogen ratio is very, very different. In aerobic processes, typically you want about 100 parts carbon per somewhere in the 10 to 5 range for nitrogen and 1 part phosphorus. And you'll notice in the anaerobic digesters, I'm looking at a wide range of something like 1,000 to 7 to 1 or 250 to 7 to 1. There's a lot of different figures you can find in literature as to exactly what these ratios should be. You will have some problems in an anaerobic digester if it is nitrogen deficient in some cases, because they do need this nitrogen for the production of enzymes and other proteins in the process. But they don't need nearly as much nitrogen relative to carbon as the aerobic microbes. And that's because anaerobic digestion as a process is much less efficient than aerobic digestion. If this were not the case, there would be lots of microbes just existing in air that were performing anaerobic digestion. That would be the favored form of metabolism, but it's not. If you've got air present, you almost always can operate a more efficient system. So because of that inefficiency, you need quite a bit more sugar input to deal with the unit of nitrogen, hence the skewed ratio. Iron and sulfur tend to make up about 0.2 in this ratio. There's a little bit of variance there. Those two are sometimes lumped into micronutrients and sometimes lumped into macronutrients. They're relevant to each other because iron and sulfur tend to precipitate out of solution, which means you need enough iron and sulfur for your system to function. If they interact, they precipitate out. Therefore, if you have high iron and low sulfur, you're going to lose all the sulfur in your system and be a sulfur deficient system. But more likely, you'll be high sulfur and low iron, and then you lose all your iron activity in the system and you end up without having any iron to use in your system. So typically you want to have quite a bit of iron in a system to make up for the sulfur present. So that's kind of how these things relate. Finally, we have micronutrients. The big four in my general thoughts are enzyme, or cobalt, nickel, selenium, and molybdenum. These are generally all assisting in enzyme processes and the production of enzymes, so they're very tied into the enzymes we'll be talking about in the rest of the presentation. If you don't have these nutrients, methanogens are unable to function efficiently. Therefore, your system really won't do anything. Following up with that, you have calcium, magnesium, copper, zinc, cadmium, all other things. There's a bunch of different metals that are all valuable, but you need such low levels that often it's just kind of like, do you have them or not? If you have them, you have enough for the system to operate properly. But yeah, this is cobalt, nickel, selenium, and molybdenum are kind of the big four. So one thing that is a challenge is identifying deficiency of micronutrients. And that's because the impacts can be a bit unpredictable. For instance, we've worked with a lot of sites I've done metals testing on, and we've observed non-detect levels of selenium in many of them. Selenium is an essential micronutrient, but if you can't detect at a low enough level to identify the selenium deficiency, you can't really make much of a statement as to whether you have enough or not. This is something we're looking into more, and we're going to start doing metals in our lab, because one of the challenges is a lot of methods for identifying metals in anaerobic digestion come from soils analysis, looking for inhibition from toxic metals like cobalt or lead. And you're not really looking for trace micronutrients in your testing. You're looking for high enough levels for them to be toxic. So if you're unable to detect them in a soils analysis, you need to have a more specialized analysis targeted for anaerobic digesters to get a good sense of if the deficiencies are real or just a detection issue. If you have a major micronutrient deficiency, you can almost completely prevent methane production, like very little will happen. But if you have a minor deficiency, your system might operate pretty much perfectly. It just might have a greater tendency to become upset and fail to metabolize volatile acids. It might suffer more if it has a slight temperature drop happen or there's a loading variance or things like that. If you introduce another form of stress on the system, the digester will be much more likely to have trouble than if you had a more optimal level of micronutrients. But there's a lot of systems you'll see have low levels of metals. They function fine for years. All of a sudden, they start seeing upsets. They don't know what's going on. And they're not going to leap to metals because intuitively, and this is often the case, if your digester operates well enough, you should have enough metals to operate the digester, right? But if you're introducing an alternative form of stress, you might have a problem. That stress could be something subtle, like you've eventually accumulated enough grit in a digester that the SRT and HRT have dropped to the point where you can no longer perform the digestion efficiently. But it also could be a case where, your mixing fails or your temperature drops a couple degrees and you bring it back up, and then you have no good way to recover the system. So you could supplement micronutrients in many of these cases if you observe a likely deficiency, so lower than optimal levels of metals, but it's not always clear cut. If it's not the main thing causing the problem, it may not make much of a difference. So keep that in mind. But yeah, addressing metals deficiencies can help with recovery following an upset as well. So the key thing, like I mentioned before, on most of these enzymes is they're heavily involved with most of the micronutrients is they're heavily involved with enzymes or enzyme activity. So if you don't have micronutrients, the microbes in the system are not going to be producing effective enzymes or enough enzymes to get the job done. So sometimes you have a case where the addition of enzymes or the addition of micronutrients could accomplish very similar things. And then you have to look to see, like, site-dependent things to see which is a better fit for your specific system. All right, so now we're moving on to enzymes. What are enzymes? Enzymes are basically tiny machines that float around in your system and randomly bump into substrate being whatever you're trying to digest, and they latch onto it and they change something about it. Generally, in practice, from what we're talking about today, they're going to break it apart into smaller chunks. There's lots of different roles for enzymes. Enzymes are basically involved in every metabolic process to some extent. So it gets a little bit too time consuming to go through every single thing associated with the enzyme processes. But keep in mind, these are tiny machines. What they do is they are proteins that catalyze or lower the activation energy of many metabolic reactions. This is very important for effective metabolic function because a lot of these applications of enzymes, you might be able to make the chemical reaction work without an enzyme, but as opposed to taking maybe like practically instantaneous reaction times where it's like a second or two, you might end up taking a week to get the chemical reaction done. You might end up taking a year. For all I know, you might end up taking a decade or 100 years to accomplish the chemical reaction without an enzyme. So the enzymes are necessary for everything to take place in anaerobic systems. Here's how enzymes work. It's a basic graphic. We got our substrate. It's going to float around and bump into the enzyme. Once it bumps into it, the substrate will stick to the active site on the enzyme. Every enzyme has a different shape active site, which is targeted for different molecules. So you can't stick to something that the enzyme is not targeted for. So they're very substrate specific. But what's going to happen is they're going to bump into the enzyme, latch onto the active site, creating an enzyme substrate complex, and you'll get an enzyme complex or a product at the end, which in our case, we're focusing mostly on the breaking apart of the different components to release them for easier digestion later in the process. Interesting thing about these active sites, there's a lot of things floating around in a digester that could bump into an enzyme that might be competitive inhibitors for the enzyme. That would mean they might be trying to stick to the active site and they might be able to work their way in and inactivate the enzyme. Enzymes can also kind of fall apart over time just naturally. That's another way the enzyme will sort of fail. They will denature. Or you can just have so many substrates, like if you latch onto substrates enough time, the enzyme will start to deform. There's also things that can stick to the wrong part of the enzyme and change the shape of the protein overall, compromising the active site. So there's a lot of different ways enzymes can be inactivated in the system, and lots of that is very relevant to the fact they are very substrate specific. They need to be exactly the right shape to work on a substrate. If they're not, the chemical reaction is not going to work. Key points for enzymes when it comes to use cases and things like that is enzymes are not alive. This is something that comes up a lot. Enzymes don't reproduce. They don't move deliberately. They just drift around in the system. They bump into a substrate that they're able to stick to. They do what they're supposed to do, and then they detach, and then they move on to look to bump to another substrate. There's nothing deliberate about it. It's just a drifting. And what that means is you need to be aware of that. You can't, like a bacteria, you can add a bacteria to a system. It is able to swim. It is able to get to a substrate. An enzyme won't do that. Enzymes do not reproduce. Eventually, they are going to fall apart in your system and no longer work. So you have an active period of time the enzyme will work in the system. Then it will eventually fail, and you'll have to add more enzyme to continue getting the benefit from adding the enzyme. Enzymes are, like I mentioned before, very substrate specific. You need to be able to have the correct enzyme to interact with the substrate. A lot of this stuff is based on the microbes in the system, which we'll talk about in the next few slides. Generally, enzymes can remain intact for as short a time as a few minutes or as long a time as a few weeks. In practice, I would say most enzymes are going to be able to remain active in a fairly environmentally favorable system for at least a day, sometimes up to a week. Enzymes are, when I say environmentally friendly systems, I'm talking about they have the correct temperature and pH requirements. Those are the main things. If you're outside of the pH or temperature range, the enzymes will fall apart more quickly and they won't be able to accomplish their jobs correctly. Enzymes, in practice, the main thing we're going to be focusing on when we talk about enzyme supplementation later is hydrolysis, the second phase in the anaerobic digestion process we mentioned before. Hydrolysis is the breaking of chemical bonds by inserting water into their structure. So a good example of that is this diagram to the right. You have a fat, a triglyceride, which I formatted just to make it a little easier to look at. It's going to break apart into fatty acids through hydrolysis, which is metabolizing this carbon-oxygen bond. You can kind of see in this little, on the top left. So it'll make these fatty acids, and glycerol is the other component from the fat that's being broken apart during hydrolysis. Other than fats, polysaccharides are going to be broken into simple sugars. Proteins are going to be broken into amino acids. This is all the hydrolysis process in action. Hydrolysis is very essential for metabolism because pre-hydrolyzed, like complex substrates, can't get through a cell membrane. They're going to be excluded by the cell, and that means they can't be used by the cell. They need to pull these substrates into the cell membrane so they can perform metabolism, use them for reproducing, use them for enzyme production later on, that kind of thing. So you need hydrolysis to take place first. Often hydrolysis is a rate-limiting step in anaerobic digestion. Typically, we're seeing either hydrolysis-limited or metals-limited system are kind of the big inhibitors. So you have your exoenzymes, which are produced from bacteria. This is excreted on the outside of the cell. They're going to be converting insoluble materials that are already disintegrated, so they're breaking down little particles in the digester into simple soluble substrates, which can be metabolized. Exoenzymes, like I said, are excreted by cells. Typically, what's going to happen is your bacterial cell might find a food substrate it wants to eat. It'll stick to it, and it'll begin excreting biopolymer and enzymes on the surface of that substrate, which are going to start this hydrolysis process. And as things like fatty acids are broken off of the structure, they're going to get transported in the inside of the cell. There's something like the keratin shuttle or something like that is going to transport them inside the cell, and then they're going to be metabolized. I have a picture from an activated sludge process that's a little slimy. Not a great activated sludge sample, but I think it's a good way to look at kind of biopolymers as they're getting produced. So you could see each of those are going to have some enzymes present in this light substance, the biopolymer, which is coating the cells. And that is going to be what they're using to break down the substrate. Now, the actual biopolymer fulfills a lot of different purposes, but one of them is to use exoenzymes to begin breaking down substrates. So now that we've talked a little bit about enzymes, we'll move on to enzyme supplementation as found in literature. There are a lot of different studies where people are using enzymes to supplement anaerobic digestion processes to prove efficacy. There are 3 examples here. The first one was talking about lipase in a grease trap. The second one is working with more of like a municipal solids kind of digester sludge. And the third one was also dealing with a municipal biosolids and applying enzymes. Now, the first one is using a single enzyme to break down a relatively simple substrate, which is grease in a grease trap. The other 2 are using blends of enzymes targeted towards what people already knew was in the waste substrate. In all these cases, people were observing a substantial increase in biogas. In the first study, it was around 30%. In the second study, we're talking about 3.65 to 5.77 times, so like a massive increase. By the way, we've done some internal studies as well, where occasionally you'll see like a 400% increase in methane generation from an enzyme. This is not normal. When you see that, it indicates you have a population of bacteria that are uniquely bad at picking up the substrate. I go on a little tangent here. We have a digester we've been operating in the lab, and we were having difficulty a few months back getting good fresh seed sludge. So we used some sludge from another study where I'd been dealing with milk waste. And we tried to apply this to a mixed synthetic substrate, which was high in cellulosic content. It was kind of funny because we were getting basically no methane generation from this cellulosic substrate. The bacteria had not figured it out at all. My guess is if I added cellulase, a cellulose degrading enzyme to it, it would have massively increased the metabolism in that digester, but we just really weren't seeing anything. At this point, we've just replaced the sludge because it just wasn't working for us there. But that's kind of the type of thing you see. And then the third study, we're seeing all the way at like an 85% increase in methane. A lot of these literature studies are looking to prove, are more of a proof of concept perspective on if the enzymes will work or not. So they're almost always focused on pre-treatment systems, and they're almost always using a very high dose rate of enzymes to ensure they're not missing any potential interaction between the enzyme and the substrate. And you can see even in these cases, they identify what the waste substrates are in the solids and break it down and add specific enzymes for each of the substrates. A lot of this isn't practical to do in a normal site, study to study, but they wanted to absolutely show if there was any potential for the enzyme to work, and they did. Pre-treatment, like I said, there's a lot of perks for pre-treatment in these enzymes applications. You have a lot of control of the pre-treatment because you can kind of do whatever you need to do if you have the equipment on site. To change the pH or the temperature constraints, you could change the holding time of your pre-treatment system, all to make sure the enzyme is functioning as effectively as possible. Whereas if you're trying to work by adding an enzyme in an anaerobic digester specifically, you have the constraint of the conditions need to be healthy for the microbes in the system. You can't be favoring the enzyme over microbes. So you have a little more constraints into what sort of enzymes you can use. You have a blend of substrates at lower concentrations in many of these cases, which means you're less likely to have the enzyme bump into the substrate. So that's why we're seeing these very high increases in methane production, particularly in study 2. A 30% increase isn't too out there for the first lipase study. In our cases, typically 20 to 25% is a pretty good result for how well an enzyme product could improve methane production in a site if it's a good fit system. I'd like to see 20 to 25%. If you're not seeing 10% or more, it's probably not a great application site. The other thing is all of these are run in BMP studies, which are like a batch loaded study. You add a bunch of substrate to a sludge on day 1, you cap everything out, and you just let it go. And sometimes it's a little hard to gauge from that how enzymes are going to interact with a real system because you have a lot of time to degrade the substrate, but you're also seeding them at the beginning with a fairly high amount of substrate. So a big slug load and then a stop. So sometimes you'll see different changes in methane production if you're comparing a BMP test to a simple system. So we've been branching away from BMP tests for studying enzyme performance. They tend to be pretty good at analyzing if a metal supplement is efficient in a site, but when it comes to enzymes, they work a lot of the time, but you aren't always sure if you see an increase, how it will pertain to the system increase. And we're trying to map that out better, but the solution in our end is moving away from that and more into a pilot study, which I'll talk about in the next few slides. So generally, literature has a lot of cases where enzymes were effective at improving methane yield. Where do you go with that? Why wouldn't they be good in my system? Why would they be good in my system? How do we really know? Enzymes are not going to be a catch-all that fits well in every system. These are some of the reasons why. The loading rates in HRT are very relevant to enzyme performance. If your digester has plenty of time to degrade a substrate without the help of enzymes, be that you have a really healthy, effective bacterial population or you just have a lot of time, you're not going to really gain any benefit out of enzymes. This is kind of like some of our sulfide digesters. We're feeding them at a lower feed rate than some of the other ones we have running. They basically run out of feed substrate in between feeds, and therefore adding enzyme isn't going to make a big methane yield difference. You're just going to see faster production for the first few hours, and then it'll slow down and match up with the control. So this isn't real valuable in a lot of cases because if you have 24 hours, it doesn't really help you to degrade all the substrate in 12 if you can take the full 24 in a lot of cases. But if you're not getting through all your substrate, the enzyme tends to be highly effective at pushing you through the substrate more quickly. The next thing would be the digester has environmental conditions which make it difficult for the enzyme to work. It could be a pH mismatch. The enzymes being selected don't fit the pH constraints of the digester. The temperature in the digester could be different from the recommended temperature of the enzyme, or there could be something that's inhibitory towards the enzyme in the digester. So you can have environmental issues with enzymes that reduce the efficacy. The nice thing about anaerobic digesters is typically they are operating at a temperature and pH range, which is fairly efficient for a wide variety of enzymes because you want them to have a neutral pH right around 7. There's a lot of enzymes that have a pH around 7 because neutral pH is very common in the environment. You may not have enough substrate present that's targeted by the specific enzyme you add. So the enzymes are going to float around bumping into things. If they don't run into the substrate they need to degrade, nothing's going to happen. This is kind of an interesting concept, but we have a lot of people asking about enzyme dose rates. When you're applying an enzyme to a site and you're thinking about the dose rate, it's not necessarily beneficial to add more enzyme to the system to deal with a higher amount of substrate. In a lot of cases, you might actually need less enzyme or about the same amount of enzyme because it's more likely the enzyme will bump into the substrate if there's more substrate present. Finally, you might have an enzyme that is not coming into the contact with the substrate effectively. A good example of this might be you have a big anaerobic lagoon and you decide, I'm going to add enzymes 50% through the lagoon over on the side. There's no mixing taking place, therefore the enzyme is not going to spread out effectively and bump into the substrate well. In that case, you'd be much better off feeding the enzyme upstream from the lagoon so it can flow in with the feed substrate and interact with as many substrates as possible. So how do you gauge an effective enzyme application? First, pay close attention to pH requirements. If you don't have a good pH requirement, your enzyme will not work as well and it will not last as long. Often, basic pHs tend to lead to faster denaturing of enzymes than acidic pHs, but you don't necessarily know unless you've looked at the spec sheet of the enzyme. With high substrate concentrations, enzymes will interact with substrate far more often. I have a study coming up with a couple slides where we talk about this a little bit more in depth. But it's kind of the concept I talked about before. If you have a high concentration of substrate, the enzymes will be able to bump into the substrate. If you have a low concentration of substrate, the enzymes may not bump into the substrate because it just won't interact. So you might actually need more enzymes for the lower concentration. Mostly mesophilic digesters are well suited from a temperature standpoint and pH standpoint for many enzymes, but you still need to double check. And be aware of other things going on in your digester before you worry about enzyme applications. A big one would be micronutrient supplementation, or if you need additional micronutrients. Basically, you need to have a healthy methanogen population for enzymes to operate efficiently because they're going to be producing more substrates for the methanogens to work with. But if you don't have enough metals or there's something else that's hindering the methanogens, you're just producing more substrate and the methanogens won't necessarily use it any more quickly. So typically, it makes a lot of sense to look at the micronutrients as well before you were considering applying any enzyme in a digester. So my concept about high substrate concentration and enzyme dose rates is pretty well illustrated here. Higher doses will always improve the degradation of substrate. If you add more enzyme, the substrate will always degrade more. But for instance, in this case, we were dosing a BMP test. We worked with the University of Oshkosh and the University of Stevens Point to compile this study. If you have a high concentration of substrate present, we were actually seeing almost the same results with 1 ppm of anazyme G versus 20 ppm of anazyme G. So the 1 ppm was able to effectively bump into the substrate and lead to the degradation. Seeing like 16,000 parts per million total fatty acids is what we were looking at in this case, going all the way down to 10,700. And then when you go up to 10 ppm, so 10 times the amount, you're only seeing a little better performance by the enzyme. So it's pretty stark. We had a very high concentration of substrates. We saw almost all of the efficacy with the lowest dose rate in this case. All right, so next thing we were looking at was our product Quiczyme L. We were comparing on different types of fat. So I included the diagram with Quiczyme L interacting with bacon grease versus Quiczyme L interacting with milk fat. These were set up identically with the same concentration of substrate. Everything was the same between the two. We were looking at total fatty acid concentration, which is a good way to measure enzyme efficacy because you're hydrolyzing fats into fatty acids. And then they're being uptaken by substrates, which in this case, we had basically a normal wastewater mixed liquor from a municipal system here as the bacterial population degrading the fatty acids. Anyway, what we were finding was after about 4 hours and 48 minutes with bacon grease, we weren't seeing that much of a discrepancy with Quiczyme L versus without Quiczyme L. Those numbers are quite close together. With bacon grease, we actually observed most of the benefits at about 10 hours into the test run, and then they got retained for the rest of the duration of the test. On the other hand, with milk fat, we were observing all of the impacts taking place in the first 4 hours and 48 minutes, and then everything basically stayed the same. Now, from a percentage of performance change, both of these looked about the same. But if you were short on time, you might find Quiczyme L was more effective on milk fat than bacon grease. So you'd want to know, oh, I at least have about 10 hours here. That means the enzyme will be effective in my system. If I didn't have 10 hours, if I was trying to get the enzyme to work in 4 hours and 48 minutes, you'd still see some result, but it won't be as good if you're working with bacon fat. So that's another thing to keep in mind with the enzymes. Another concept I mentioned a couple times earlier on, but I want to elaborate on, is if you have an enzyme limited system, so a system that's failing to perform hydrolysis, or if you have a micronutrient limited system, so there's not enough micronutrients for the methanogens to function efficiently. So here's two examples. I'm comparing a dairy manure BMP test with a slaughterhouse wastewater gas production test. This was done by Digester Doctor Labs. We can see with biogas 1 in the dairy manure test, we actually observed the bulk of the benefits with biogas 1 by itself, and we found that the addition of Quiczyme P at this time didn't make that big a difference. We were just observing a small bump in methane production after that point. The biogas 1 did most of the work. That is a pretty clear example of a metals limited system. If you have a metals limited system, which you'd find out by screening for metals or do some sort of BMP test or another analysis to confirm that metals are beneficial. You can figure that out. Your enzyme is only going to make a little bit of a difference, and you'll need to supplement the metals first to see the impact. Whereas on the right, we're looking at a slaughterhouse wastewater sample for gas production, and what we're seeing is biogas 1 by itself did not make much of a difference at all. This would be a clear hydrolysis limited system. Once we added Quiximel and bug juice, in this case, is what digester doctor was working with, we saw a dramatic increase in methane production there. So it's a case of one of the additions helped in the first one and one of them helped in the other one. That's why I recommend doing metals analysis prior to considering any of these additives in your system. You need to know what is actually rate limiting in your system to be able to effectively apply a product. All right, so another example, I was observing a 22% increase in a dairy manure digester with the addition of Enzyme P at 5 ppm. When we added metals by itself, it made no difference, but metals plus Enzyme with Enzyme P made a little bit of an improvement. Like we went from 1509 to 1570, adding metals to the Enzyme P. But the Enzyme P itself led to a 22% increase in biogas, which to me is a fairly, it's pretty clear the enzyme is working in that case. So here, we would not see a lot of benefit from adding the metals there. However, in the same system, we noticed with Enzyme G, a different blend designed to target fats, we were actually seeing a much bigger difference with the addition of metals, which you can see looking at the rate of methane production over 6 to 31 days in the study. You can see Enzyme G with added micronutrients was producing methane almost 50% faster for a large chunk of that time. And then eventually from 25 to 30 days, we were seeing Enzyme G without metals catch up and pass up the Enzyme G nutrients and methane production rate. But it ended up making a much more sizable difference adding metals with Enzyme G versus metals with Enzyme P. So just looking at these two sites, you can see one of them was more metal limited. When we were hydrolyzing proteins, we were mostly dealing with a hydrolysis limitation. When we were trying to degrade fats, we actually were dealing with more of a nutrient limitation than an enzyme limitation. So same site, different hydrolysis aid, and you saw different results that way. I've been dealing with a lot of BMP studies over the past year. We run some in-house. We've worked with a number of different sites to run BMP studies for us to study all this literature. Most of the academic studies that are done on enzyme supplementation are done in BMP tests. They're pretty standard. But like I said before, you have a big problem with organic loading and substrate concentration in BMP tests, which can make it difficult to effectively gauge if an enzyme is effective in a system. Generally speaking, you'll see the result you predict, but you won't have a good sense of what % increase you'll see in real use case. So it's not operating like a real system, which means while you may know if in practice the enzyme is effective in a site, it won't actually help you gauge if it is worthwhile to do that or not. You'll have to do a full-size site trial in this case to figure that out. So because of that, we developed our pilot digester method. Right now we've got about 15 pilot digesters going. It's getting a little crazy over here since we just started operating them all. We've got 4 digesters working with dairy manure. Right now we're studying our product Enzyme D, and we're using metal supplements as well. We're working with Enzyme P and hydrogen sulfide production in several of our digesters to study that relationship, and we're also working with Enzyme G, attempting to accelerate the degradation of dairy wastewater as well. So that's all going on right now, but basically the idea of this study is they're loaded with site sludge and then fed 3 to 5 days per week, depending on the requirements of the study, with feed substrate. So they're fed at a rate that's proportionate to a real anaerobic digester, which helps us get a lot closer to replicating appropriate loading, and we're tracking all the data over time to see what things meaningfully will make a difference for a digester site versus what is conceptually going to help but might not matter depending on how site operation is going. We're controlling pH, HRT, and organic loading and that kind of stuff to give as much control over variables as we can, and making sure there's no oxygen at the beginning of the study, that kind of thing. So that's how that process generally works. So the first pilot study I wanted to talk about was a study on Enzyme P and hydrogen sulfide reduction. We were focusing exclusively on biogas quality, so we're collecting all the biogas in gas bags and running it through a gas meter to analyze hydrogen sulfide content versus methane and carbon dioxide, that kind of thing. But we've noticed in a number of BMP tests that Enzyme P seemed to lead to some reduction in hydrogen sulfide, and we wanted to expand on that and better understand what's going on, because we don't really understand initially why that would be taking place. Like, we have some theories, but we want to understand it better. So the feed we used in this case was a mixture of milk and cream, which is easy to supply consistently. So it made it a good fit for the study. It's also a fairly representative feed substrate for looking at something like a dairy digester. This study was a follow-up to work we did after establishing our effective feed blend to produce adequate hydrogen sulfide for our test, and we also modified our HRTs to get the appropriate hydrogen sulfide growth in previous studies. So we had a sense of how we wanted to operate this, which was a HRT of 22 days and a feed rate of 40 milliliters of milk with a 1% additional cream addition for each of the feed substrates. So looking into that, we also had Enzyme G reactors, which we did not believe would make any difference in hydrogen sulfide reduction, and a control with no enzyme as a comparison here. So first off, they all got the same amount of feed. This is not a very exciting graph because there's no way to tell anything apart. Everything is operating identically. And this is what you'd expect because we weren't pushing the limits of organic loading in this case. We were just trying to feed them a modest feed with enriched sulfate so it would produce hydrogen sulfide so we could go on from that. So we can see all the digesters were operating the same in terms of methane and biogas volume. Now, in this study, we operated it for 6 weeks prior to the supplementation of any enzymes to give us a solid baseline for the production rate, and you'll notice we were trending upward in hydrogen sulfide production for the first 6 weeks here with Enzyme G and our control digesters. And we had the sulfides continue to increase. There was a little dip at week 10. This was unfortunately during the holidays, so we weren't able to get them quite as many feeds as we were normally able to in the week, but then you can see it trend up again after that. So this is kind of how we expected the controls to behave. We're having some variance in hydrogen sulfide, but you can see these ones are all behaving fairly predictably. However, when we contrast that with Enzyme P in our two Enzyme P digesters, which are A1 and B2, we can see the sulfides increased for the first 6 weeks, just like the other digesters, but when we started adding Enzyme P to the system, we noticed the slope reversed, and the hydrogen sulfide concentrations were going somewhat consistently downward throughout the rest of the study. So you're looking at the levels. None of these are like really extreme cases for hydrogen sulfide. They're pretty modest, but we're seeing something like 500 versus 600 there versus 600 to 700. So it seemed to be a fairly notable case where we were seeing a downward slope versus the other test run with Enzyme G in the control, where we were not seeing a clear downward slope after that point. Now, it's not as distinctive as we'd like, and this is still in progress because we're increasing the sulfate concentrations to see if we strain the system more, if this trend will continue, or if it's basically stuck, like if it's just going to flatline at a slightly lower level. We need to understand that kind of thing. But it was an interesting anomaly we were able to observe because of doing this new pilot system setup. So here's a reference on the slopes. You can see both the Enzyme P1, or so A1 and B2, the Enzyme P reactors, started out with a positive slope from weeks 0 to 8. And then from 8 to 13, they had a decrease in slope, whereas the control and the Enzyme G, there was still an upward trend at that point. So why did the hydrogen sulfide seem to decrease here? It's really hard to say. We've observed with Enzyme P a number of cases where we see an increase in methane quality and a decrease in hydrogen sulfide, but we don't necessarily see an increase in methane in some of these cases. Sometimes you do, but not often. So what is happening? Currently, the main theory we have, which we have to do a lot more work to verify if this is correct or not, but the main thing is you're releasing more free amino acids, which are able to be uptaken by other microbes in the process. And it's creating a change in the competitive environment slightly, which seems to be favoring other organisms over sulfur-reducing bacteria, leading to a reduction in sulfate. Because keep in mind, they're all getting the same sulfate loading over time. Unfortunately, at this point, there's some discrepancy between the replicates and that kind of thing, which all needs to be worked out through a longer study run. So I can't make a more definitive conclusion than that, but that seems to be what makes the most sense. We're not adding very much anazyme G or anazyme P, and we compared it to anazyme G, which from like a carbon loading standpoint is very similar. That doesn't seem to make any meaningful difference. Like I said, we're continuing on this study with higher loading rates and more sulfate to see if this trend continues. So the next thing we did was an anazyme G pilot study. The digesters were operated at 36 C and maintained at pH 7.3 for the duration of the study. We were feeding them a similar feed to the Anazyme P study we were just running, but we increased the loading rates to be quite a bit higher because we were hoping to strain the system enough that it wasn't able to get through the feed substrate between feeds. So we'd actually see if there was a meaningful increase in gas production in that environment. So we're measuring biogas production rates in this case with gas flow cells. And Anazyme G was applied at a fairly high dose rate of 20 ppm in this case, because we wanted to have as distinctive results as possible. So this is how it kind of worked. For reference, I'm showing the graph here for 60 milliliters of milk feed and 80 milliliters of the milk feed. So we have a about a quarter increase in loading between the two. And we actually did start off initially with a 40 milliliter increase in milk feed, and we're tracking it. And we were seeing like a 1 to 3% increase in methane generation to about 20 hours, and then they would level off there. So we weren't really loading the system heavily enough for there to be any strain. But when we increased to 60 milliliters of milk feed, we noticed that we were actually maintaining a higher methane production rate all the way to about 35 hours in this study, at which point the control actually produced more because Anazyme G ran out of substrate. So we were kind of approaching our goal for straining the system. When we went up to 80 milliliters of milk fat, the control stayed behind Anazyme G for the entire duration of the test, which would be more like a heavily loaded system. You still have some substrate left over between feeds. And that's where you seem to get a prime case where it is beneficial to add enzyme, as opposed to the previous case where if you had 40 hours, wouldn't really matter very much what happens. You'd still be getting the same degradation rate. Another note, which is kind of a fun note, I increased the milk feed up to 120 milliliters after this, and the control digester just completely crashed right away, whereas our test digester did survive the loading rate. So it didn't seem like we were increasing the strain on the methanogens with the addition of the enzyme there. It was more a case of it seemed to be having less accumulation over time between feeds, which allowed it to hang in there a little longer. That's a pretty extreme loading rate, so I think we ended up having to pause the test because I really didn't want the control digester to die. I wanted it to keep operating because I wanted to study the discrepancy longer, but it crashed, and I don't know what would happen with the Anazyme G if I kept loading it at that rate, but I ended up blending them together, restarting them, and getting everything to work again to fix the problem. So this was just comparing one digester to a control. I've got it replicated now because I don't want all my digesters to crash again. I get these sort of things popping up in research and development. Sometimes things don't go quite as planned, but in general, we're still seeing this kind of trend going on in the new digesters. So this test is also still ongoing. In fact, both of these studies have got about a month left before we have the full literature done. We're probably going to release some papers on this sort of thing later this year if people have any interest. So let us know if you're interested in receiving that kind of paper. Helps me prioritize what sort of things I focus on. But the main takeaway here is if you don't have a high enough loading rate, it doesn't matter if you add the enzyme because your control digester still has enough time to function properly. Whereas if you're able to increase the loading rate, you start to see a discrepancy. Like in the graph above, I was seeing about a 14% increase in methane. And if I went up to 90 milliliter of cream feed, I would likely have seen a slightly larger methane increase. You get this kind of case where you're starting to stress the system out and you can't go too far, because if you go too far, the digesters will die. But if you don't go far enough, then you're not able to see any discrepancy other than tracking the rates to see about how long it takes for the two digesters to match up. All right, so to review everything, enzymes are proteins, they're machines, they move around and bump into substrate to break them down. They're necessary for most metabolic functions. Generally speaking, micronutrients are required for enzyme production and use. Micronutrients also work for processes in anaerobic digesters, which might decide whether your system is enzyme limited or micronutrient limited. So you have to watch out for micronutrients as well with their relation to enzymes. Sometimes they're much more effective if they're both in the system at the same time. Sometimes you can add one or the other and you'll see benefits that way. Exoenzymes are the enzymes that promote hydrolysis. These are produced by many different microbes, but generally this is what you're doing when you're adding an enzyme supplement, is you're effectively adding exoenzymes to the system to break everything down. And the keys are, if you're working with an enzyme, make sure it's well suited for your system. So you have an enzyme that matches up with substrate, pH, and temperature requirements for the site. You're going to also want to make sure that your digester is operating effectively other than that. Like, your digester should be doing okay for it to benefit from seeing an enzyme. If your digester is completely failing because the pH is 6 or your mixer is not working or it's operating at 20 degrees cooler than ideal, these are extreme examples. But if your digester has a massive pressing problem, adding enzymes is not going to do anything to the system. You have to make sure it's a somewhat acceptable environment in there first. So that's the kind of key things you need to keep in mind for the enzymes. All right, so reiterating from earlier on, if you have any questions or concerns about your wastewater system, anaerobic or aerobic process, feel free to reach out to Aquafix Labs. We do a lot of treatability work. We do microscopy, that kind of thing. We're doing a lot of the study and development on products, and we're always more than happy to answer questions on applications and how to make sure any product application is working as well as possible. We'll also just work with you on general site troubleshooting if you're just trying to figure out a long, stubborn problem. So please just reach out to us if you have any questions or want any help with your wastewater process from our lab services. All right, well, here are all my works cited in the presentation. If you have any questions about any of the research or literature associated with this presentation or have any more in-depth questions, please feel free to reach out to me. Thank you all for attending today's webinar. If you're interested in seeing any of our future webinars, feel free to sign up for our newsletter and we'll reach out to you with any new content coming up.